Hello and welcome to PharnaTelevision News Review at the World Stem Cell and Regenerative Medicine Conference 2015 in London. On today's show I have Chris Parker of Cellular Dynamics. Chris, welcome.
Thank you. Thank you for having me.
So, Cellular Dynamics, you've got a couple of interesting products about that. Perhaps you could tell us a little bit about the company and the products.
Sure. Well, we're one of the first iPS, induced pluripotent stem cell-based companies. We're the largest producer of iPSCs in the world, and we have a variety of different products that we can manufacture from iPSCs that represent a variety of tissues in the body, things like cardiomyocytes, neurons, hepatocytes, that have been used for drug discovery research, safety testing, but also have a great deal of therapeutic potential.
So for the layman, if you like, you can take any cell type from somebody, a blood draw, and turn them into any other tissue.
Exactly. Literally take a standard doctor's office blood draw, in a vacutainer, have it shipped to us. We can then take the cells out of that, turn them into iPSCs over time, and then use that as a master cell bank to produce virtually any cell type in the body.
So, and these cells are then primarily used in research, or do you see them, will they be used in clinical utility as well?
Well, our first entry into the market as a startup company to generate revenue was to invigorate the research tools market. There was an unmet need for human cells in the drug discovery process. And our founder, Jamie Thompson, who was one of the first inventors of human embryonic stem cells and was also co-published with Dr. Shinya Yamanaka on iPS cells at the same time, felt like although cell therapy was going to be important, having access to human tissue was also going to be critical to provide safer and better, more effective medicine. So we focused on industrialization to be able to produce large quantities of these cells so that they could be used for drug safety testing and for high throughput screening for diseases of interest.
It's interesting you mentioned high throughput screening there, because that was hailed as the technology is going to bring a whole new suite of drugs using cell banks and cell lines, immortal cell lines that were more relevant to disease models, and it didn't quite materialize. What's going to be different with stem cells?
Well, these are the true human authentic cell. They're post-mitotic, and they represent all the attributes of an endogenous cell. They're not a cancerous cell line that's been transformed with some gene manipulation to quote-unquote look like a cell type of interest, right? So our cardiomyocytes, for instance, beat spontaneously in culture. You can do contractility studies, you can look at arrhythmias. That was unheard of in an in vitro system. So in a lot of ways, the technologies for cellular analysis, imaging and things like that, had outpaced the biology that pharma had been using previously. And so now having the authentic cell allows really to make all of those platforms better and actually get results that are more relevant to the human. In addition to that, we can actually put the clinical trial into the dish. We can represent your and my differences, either from a disease basis or an ethnicity basis or any other difference that we might see in terms of response to drugs, and actually replicate that in the dish.
So have you kind of flipped it on its head then? Because previously it was a one cell fits all. Would you now have to do multiple different patient types, if you like, and derive the stem cells from them and then test those to get a realistic picture?
Well, that's indeed what we do, right? I mean, the whole idea is to we call it the in-vitro clinical trial, right? Instead of doing all my discovery work on one cell type that's supposed to represent everybody, even though we inherently know that it won't, we now can actually take that patient and put it into the dish so we can understand before we do our candidate selection that we're actually representing a drug that's going to be effective over a diverse set of population as opposed to the perfect patient.
And can you take it beyond the drug discovery phase? So you could actually, I'm thinking there are savings here in animal testing, in clinical testing, in humans.
No question.
And it could do that as well?
In fact, there's an example of that right now. There was where there's a program that's been sponsored by the FDA called CIPA, the Cardiac In Vitro Proarrhythmia Assay Initiative. And this is really an initiative to eliminate some of the draft guidance that causes pharmaceutical companies to do what they call a thorough QT trial. This is a trial that is really a null hypothesis. It's a trial that has healthy, normal people that take a small dose of a drug to see whether they get any kind of arrhythmia, right? Costs about $2 million, 60 patients, and with the whole result being didn't do any damage, didn't cause any heart arrhythmias. So the guidance now is to test our cells along with some bioinformatic tools and imaging tools to be able to see if we can replace that trial by doing it in vitro, thus eliminating a need for a given required clinical trial.
So as well as getting the right target and the right mechanism, there are cost savings in the process. There should be time savings, regulatory benefits, and better drugs to market quicker. That's the ultimate goal.
That's our pitch. And the beauty of it is the data is speaking for itself. We've tested literally hundreds of drugs now to show that we can eliminate false positives and false negatives that are typically seen in animal models. We've demonstrated that our cells are actually more physiologically relevant and sensitive compared to animal models. We had actually one example with a pharmaceutical company where they had a drug that had passed all their in vitro tests, passed their animal tests, and got into clinical trials. And retrospectively, they took that drug because it turned out that the patient was an order of magnitude more sensitive than the animal model that they had used. They took that drug, brought it back, and put it onto our cardiomyocytes, and the cells responded exactly as the patient would have. So they could have eliminated that entire animal study and killed that drug early, thus saving literally millions of dollars if they would have used our cells first. And now that particular company tests all their drugs that are going into animal models on our cardiomyocytes.
So you are truly enabling the fail fast, fail cheap, but also on top of that, you're doing the succeed faster with better drugs.
Exactly, with data.
With data. Excellent. So presumably, ultimately, beyond the mimicking clinical trials and doing them in a better way, there are therapies in what you have. You'll be able. There are cellular therapies there.
Yeah, no question. I mean, by creating and having the ability to robustly and under very controlled requirements, producing industrial quantities of cells, and with the scale and under controlled systems, we can actually have now been able to put these cells into key opinion leaders' hands to really determine if they have therapeutic efficacy, therapeutic effect. And so from the tools business, we're actually evolving now into a therapeutics business by creating authentic cells that can be used in a variety of different disease areas. The three main programs that we're focusing on are Parkinson's disease, myocardial infarction, and age-related macular degeneration.
Right, so some pretty big disease areas.
Large
there, yes.
unmet needs, huge markets. Really a degenerative disease, we believe, requires a curative type therapy. These are diseases that the population is growing as our age continues to be expanded, and therefore having a curative therapy that not only potentially, like for instance with age-related macular degeneration, prevents further blindness, but one that potentially could restore vision is truly revolutionary.
So, given you've described some research tools type approaches and also some new drug approaches, new therapy approaches. What's the business model that goes with that? Is it fee for service or what is it?
Well, the business model is to create off-the-shelf products that are cryopreserved, that can be shipped at any time to a customer. So our business started out with a generic cell type from a single iPS line that we call iCell, because they're derived from iPS cells. Then we evolved into a program called MyCell, which is giving our customer pharmaceutical company the ability to just simply send us blood samples from donors from a clinical trial, for instance, that then allows us to create the iPS cells and then the target cell of interest, and then ship it back to them under the same quality systems that we ship our iCells. And we have a number of examples where we've done projects for big pharma in that area.
So will you look at any time to, you talked about those disease areas, to reach through into actual therapies and generate
At this point,
revenues?
we make enough money on the actual delivery of the cells to not have to have that discussion. It usually, in a lot of ways, is a game stopper from a negotiation standpoint, because we know that there's much more to getting a drug to market than simply using our tools. And as long as we can be profitable based on the margins that we make in our manufacturing process, we can be a viable business.
And clearly that's an attractive proposition, because recently your company, following a successful IPO, you were bought by Fuji.
Yes, by Fujifilm. We've finally been able to become more Japanese. Because the Japanese market, as you can imagine, with the regulatory benefits afforded to those that are seeking to do iPS therapies in Japan. Being an outsider to that made it a little bit difficult for us. And now having a benevolent owner in Fujifilm, we now can participate in those activities and truly help Japan go faster.
And Fujifilm clearly known for its film business from way back, but also we know it's got involved in biologics and producing proteins and antibodies. This seems a little bit different. Is Fuji going to become a therapeutics company?
Well, they already are, actually. Right. They have Toyama Chemical Company, which is their pharmaceutical division, kind of small molecule, typical pharma. They also have Fujifilm Regenerative Medicine, which is a division that's focused on aspects related to cell therapy. They also bought a company called Japan Tissue Engineering Corporation, which has actually the first 2 cell-based therapies that have been approved in Japan. Those are adult stem cell-based therapies, but our the kind of sponsor in Mr. Toda, who is also the chairman of FIRM in Japan, which is the Forum for Innovative Regenerative Medicine, which encompasses all major organizations in Japan and academia. He saw the kind of the value of being able to buy the leader in the field and have Fujifilm really jump to the head of the line in terms of engaging in iPS-based cell therapies.
So it seems like a very astute move by Fuji to acquire your business.
Well, I would agree. And it was beneficial to our shareholders, so I think they were happy too.
Indeed, and we should expect to see a lot of therapies make it to the market soon with cellular dynamics inside, if you like.
That was the goal from the very beginning, so I appreciate you acknowledging that marketing concept.
Well, we'll look forward to hearing and seeing more about that. Chris, thank you very much for joining us today.
Thank you. Thanks for the time.